Good morning, my name is Jado, Laredo, and I will be discussing nucleic acid. So, nucleic acids are the molecules within a cell responsible for containing instructions needed for making the complete organism of which they are a part. It was discovered by Swiss physiologist Friedrich Mischer in 1869 while studying the nuclei of the white blood cells. So, nucleic acid have two types, the DNA and RNA. It was not until 1944 that scientists have obtained the first evidence that the nucleic acids are responsible for the storage and transfer of genetic information. And all nucleic acids are polymers in which the monomer units are nucleotides. So, type of nucleic acids. The DNA functions primarily on storage and transfer of genetic information, which is used indirectly to control many functions of a living cell. Nearly all of the DNA is found within the cell nucleus, and DNA is passed down from existing cells to new cells during cell division. While an RNA functions primarily in the synthesis of proteins, molecules that carry out essential cellular functions. And lastly, RNA occurs in all parts of a cell. So next is the nucleotides. Nucleotides are the building blocks of nucleic acids. To understand what this means, they are simply related to nucleic acids in the very same way that amino acids are related to proteins. So a nucleotide is a 3-subunit molecule in which a pentose sugar is bonded to both a phosphate group and nitrogen-containing heterocyclic base. So, more than complex monomers than monosaccharides of polysaccharides and amino acids of proteins. So the three parts of a nucleotides are the pentose sugar, the phosphate group, and the heterocyclic bases. So the three parts of a nucleotide is arranged in this manner as shown in the diagram. Pentose sugars, or the sugar unit of a nucleotide, is either the pentose, ribose, or the 2'-deoxyribose. Structurally, the only difference between these two sugars occurs at the carbon 2'. The hydroxyl group present on this carbon is in ribose becomes a hydrogen atom in 2'-deoxyribose. That's why the prefix deoxy means without oxygen. One thing to note is that the system for numbering the atoms in the pentose and nitrogen-containing base subunits of a nucleotide are important and will be used extensively in later sections of this video. The convention is that first, pentose ring atoms are designated with primed numbers. And second is nitrogen-containing base ring atoms are designated with unprimed numbers. Next is the phosphate. The phosphate group is derived from phosphoric acid. Under cellular pH condition, the phosphoric acid loses 2 of its hydrogen atoms to give a hydrogen phosphate ion. Next is the nitrogen-containing heterocyclic base. These bases are nucleotide components. 3 of them are derivatives of pyrimidine, a monocyclic base with a 6-membered ring, and 2 are derivatives of purine, a bicyclic base with fused 5 and 6-membered rings. These bases contains amine functional groups, the secondary or tertiary, which make it have basic characteristics. In terms of nucleic acid differences, adenine, guanine, and cytosine are found in both DNA and RNA, while uracil is found only in RNA and thymine usually occurs only in the DNA. Next is the nucleotide formation. The formation of a nucleotide from sugar, base, and phosphate can be visualized as occurring in the following manner. Important characteristics of this combining of 3 molecules to form into a nucleotide are that condensation or the formation of water molecule. Second is the base is always attached at the C1 position of the sugar. For purine bases, attachment is through the N9, and for pyrimidine bases, N1 is involved. The carbon 1' carbon atom, the C1' carbon atom of the ribose unit is always in a beta configuration, and the bond connecting the sugar and base is a beta and glycosidic linkage. The phosphate group is attached to the sugar at the C5' position through a phosphate ester linkage. Next is nucleotide nomenclature. There are 4 possible RNA nucleotides differing in the base present A, C, G, or U, and 4 possible DNA nucleotides differing in the base present A, C, G, or T. The common name and abbreviations for the 8 nucleotides of DNA and RNA molecules are below. It is important to be familiar with them. We can make several generalizations about the nomenclature given in the table. First is that all of the names end in 5' monophosphate, which signifies the presence of a phosphate group attached to the 5' carbon atom. And then second is preceding the monophosphate ending is the name of the base present in a modified form. The suffix "-ocene" is used with purine bases, and the suffix "-idine" is used with pyrimidine bases. The third one is the prefix deoxy at the start of the name signifies that the sugar present is deoxyribose. When no prefix is present, the sugar is ribose. Lastly, the abbreviations for the nucleotides come from one-letter symbols for the bases, A, C, G, T, U, the use of MP for the monophosphate, and a lowercase d at the start of the abbreviation whenever deoxyribose is the sugar. Next is primary nucleic acid structure. Nucleic acids are polymers in which their repeating units, the monomers, are nucleotides. The nucleotide units within a nucleic acid molecule are linked to each other through sugar-phosphate bonds. The resulting molecular structure involves a chain of alternating sugar and phosphate groups with a base group protruding from the chain at regular intervals. The alternating sugar-phosphate chain in a nucleic acid structure is often called the nucleic acid backbone. The backbone is always an alternating sequence of phosphate and sugar groups, where the sugar is ribose in RNA and deoxyribose in DNA. And one thing to note is that primary nucleic acid structure is the sequence in which nucleotides are linked together in a nucleic acid. Because the sugar-phosphate backbone of a given nucleic acid does not vary, the primary structure of the nucleic acid depends only on the sequence of bases present. Just as the order of amino acid side chains determine the primary structure of protein, the order of nucleotide bases determines the primary structure of nucleic acid. We can now define in terms of structure the 2 major types of nucleic acids. The DNA, which is a nucleotide polymer in which each of the monomers contains ribose, a phosphate group, and one of the heterocyclic bases, adenine, cytosine, guanine, or uracil. Then the DNA, which is a nucleotide polymer in which each of the monomers contains deoxyribose, a phosphate group, and one of the heterocyclic bases. Important points about the nucleic acid structure is that first, each non-terminal phosphate group of the sugar-phosphate backbone is bonded to the 2 sugar molecules through a 3'5' phosphodiester linkage. There is a phosphoester bond to the 5' carbon of one sugar unit and a phosphoester bond to the 3' carbon of the other sugar. Second is a nucleotide chain has directionality. One end of the nucleotide chain, the 5' end, normally carries a free phosphate group attached to the 5' carbon atom. The other end of the nucleotide chain, the 3' end, normally has a free hydroxyl group attached to the 3' carbon atom. By convention, the sequence of bases of a nucleic acid strand is read from the 5' end to the 3' end. And lastly, each non-terminal phosphate group in the backbone of a nucleic acid carries a -1 charge. So, just like in proteins having an N-terminal end and a C-terminal end, nucleic acids have a 5' end and a 3' end. So the 3 parallels between primary nucleic acid structure and primary protein structure are worth noting. DNAs, RNAs, and proteins all have backbones that do not vary in structure. The sequence of attachments to the backbones, nitrogen bases in nucleic acids, and amino acid R groups in proteins, distinguishes one DNA from another. The same as well for one RNA from another and one protein from another. Both nucleic acid polymer chains are And protein polymer chains have directionality. For nucleic acid, there is a 5' end and a 3' end. And for proteins, there is an N-terminal end and C-terminal end. So next is the DNA double helix. Like proteins, nucleic acid have secondary or three-dimensional structure as well as primary. The secondary structure of DNAs and RNAs differ, but we will discuss them separately. The amount of bases A, T, G, and C present in DNA molecules were the key determination of the general three-dimensional structure of DNA molecules. Base composition data for DNA molecules from many different organisms revealed a definite pattern of base occurrence. The amount of A and T were always equal, and the amounts of C and G were always equal, as were the amounts of total purines and total pyrimidines. The DNA double helix involves two polynucleotide strands coiled around each other in a manner somewhat like a spiral staircase. The sugar-phosphate backbone of two strands can be thought as the outside banisters of the spiral staircase. The two strands are connected by hydrogen bonds between their bases. The bases, or the side chains of each backbone, extend inward towards the bases of the other strand. Additionally, the two strands of the double helix are antiparallel, which means that they run in opposite directions, one strand in the 5 to 3 direction and the other in the 3 to 5 direction. Three views of the DNA double helix. A schematic drawing that emphasizes the hydrogen bonding between bases of the two chains. The next is a space-filling model in which the strand is red or orange and the bases are shown in lighter shades of blue and white. The last one is a top view of the double helix. Next is base pairing. The physical restriction, the size of the interior of the DNA double helix, limits the base pairs that can hydrogen bond to one another. Only pairs involving one small base, a pyrimidine, and one large base, a purine, correctly fit within the helix interior. There is not enough room for two large purine bases to fit opposite each other, since they overlap, and two small pyrimidine bases are too far apart to hydrogen bond to one another effectively. Of the 4 possible pyrimidine combinations, hydrogen bonding possibilities are more favorable for the A, T, and G, C pairing. So, this picture shows the specific hydrogen bonding interaction for the 4 possible pyrimidine base pairing combinations. As you can see, the thymine-adenine base pairing and the cytosine-guanine base pairing have 2 and 3 hydrogen bonds in between the bases. And note that these two have the highest number of hydrogen bonds that have formed. So next is an exercise. If a template strand of DNA is organized in the 5' CGAAT, CCTA 3' order, then what is the complementary DNA strand that is created from this template during replication? And the answer for that would be letter C. So base stacking interactions. Stacking interactions involving a base and the parallel bases directly above it and below also contribute to the stabilization of the DNA double helix. So next is replication of DNA molecules. DNA molecules are the carriers of genetic information within a cell. They are the molecules of heredity. Each time a cell divides, an exact copy of the DNA of the parent cell is needed for the new daughter cell. The process by which new DNA molecules are generated is DNA replication. DNA replication is the biochemical process by which DNA molecules produce exact duplicates of themselves. Note that the key concept in understanding DNA replication is the base pairing associated with the DNA double helix. So to understand DNA replication, we must regard the two strands of the DNA double helix as a pair of templates or patterns. During replication, the strands separate under the influence of the enzyme DNA helicase, and the hydrogen bonds between complementary bases are broken. This unwinding process is somewhat like opening a zipper. Each can then act as a template for the synthesis of a new complementary strand. The The base of the separated strands that are no longer connected by hydrogen bonds can pair with free individual nucleotides present in the cell's nucleus. The base pairings always involve C pairing with G and A pairing T. The pairing process occurs one nucleotide at a time after a free nucleotide has formed hydrogen bonds with the base of the strand. The template strand rather. The enzyme DNA polymerase verifies that the base pairing is correct and then catalyzes the formation of new phosphodiester linkages between the nucleotide and the growing strand. The DNA polymerase then slides down to the strand to the next unpaired base of the template, and the same process is repeated. The enzyme DNA polymerase can operate on a forming DNA daughter strand only in the 5' to 3' direction because the strands of the parent DNA run in opposite direction. One is 5' to 3' and the other is 3' to 5' direction. Only one strand can grow continuously in the 5 to 3 direction, and the other strand must be formed in short fragments called Okazaki fragments. The breaks or the gaps in this daughter strand are called nicks. To complete the formation of this strand, the Okazaki fragments are connected by action of the enzyme DNA ligase. The result is two daughter DNA molecules with base sequences identical to those of the parent double helix. The process of DNA unwinding does not have to begin at the end of the DNA molecule. It may occur at any location within the molecule. Study shows that unwinding usually occurs at several interior locations simultaneously, and that the DNA replication is bidirectional for these locations, since it proceeds in both directions from the unwinding sites. As shown in the picture, the result of the multiple site replication process is the formation of bubbles of newly synthesized DNA. The bubbles grow larger and eventually give rise to the two new daughter DNAs. Also note that the multiple site replication process enables large DNA molecules to be replicated rapidly. So next is chromosomes. The DNA molecule is a few centimeters long, but just a couple of nanometers wide. So to prevent the DNA from beginning to becoming a tangled mess, they are wrapped around proteins called histones, forming a nucleosome. These nucleosomes are bundled together into a fiber called chromatin, which is further looped and coiled to form a chromosome. A complete summary tells us that once the DNA within a cell has been replicated, it interacts with specific proteins in the cell called histones to form structural units that provide the most stable arrangement for the long DNA molecules. It is important to know that humans have 23 pairs of chromosomes for a total of 46. Next is protein synthesis. We saw in the previous topic how the replication of DNA makes it possible for a new cell to contain the same genetic information as its parent cell. We will now consider how the genetic information contained in a cell is expressed in a cell operation. This brings us to the topic of protein synthesis. The synthesis of proteins such as skin, hair, enzymes, hormones, and so on is under the direction of DNA molecules. It is the role of DNA that establishes the similarities between the parents and the offspring that we regard as hereditary characteristics. We can divide the overall process of the protein synthesis into two phases. The first phase is called the transcription, and the second is translation. The following diagram summarizes the relationship between transcription and translation. Before discussing the details of transcription and translation, we need to learn more about RNA molecule. They are involved in both transcription as the end products and translation as the starting material. We will be particularly concerned with the differences between RNA and DNA and among various types of RNA molecules. So, ribonucleic acids and its major differences between the RNA molecules and the DNA molecules. So first is the sugar unit in the backbone of RNA is ribose and deoxyribose in DNA. The base thymine is found in DNA and it is replaced by uracil in RNA. In RNA, uracil instead of thymine pairs with adenine. Then third is RNA is a single-stranded molecule, whereas the DNA is double-stranded. Thus, RNA, unlike DNA, does not contain equal amounts of specific bases. Then lastly, RNA molecules are much smaller than DNA molecules, ranging from 75 nucleotides to a few thousand nucleotides. Note that the single-stranded nature of the DNA does not prevent portions of an RNA molecule from folding back upon itself and forming double helical regions. If the base sequences along two portions of an RNA strand are complementary, a structure with a hairpin loop results. Next is the type of RNA molecules. This can be categorized into 5 major types, distinguished by the function. These 5 RNA types are the hRNA, the mRNA, the snRNA, the rRNA, and the tRNA. So the hRNA, or heterogeneous nuclear RNA, is formed directly by transcription, and post-transcription processing converts the heterogeneous nuclear RNA to messenger RNA. This type of RNA make up about 5% to 10% of the RNA population. Next is messenger RNA. Messenger RNA is RNA that carries instructions for protein synthesis to the sites for protein synthesis. The molecular mass of the messenger RNA varies with the length of the protein whose synthesis it will direct. Then next is the small nuclear RNA. This is the RNA that facilitates the conversion of heterogeneous nuclear RNA to messenger RNA. It contains from 100 to 200 nucleotides. The next is ribosomal RNA. It is an RNA that combines with specific proteins to form ribosomes, the physical sites for protein synthesis. The rRNA present in ribosomes has no informational function, and it comprises about 75 to 80%. And lastly is the transfer RNA. They are RNA that delivers amino acid to sites for protein synthesis. Transfer RNAs are the smallest of the RNAs, possessing only 75 to 90 nucleotide units, and they only comprise 10 to 15% of the population. So, at a non-detailed level, a cell consists of a nucleus and extranuclear region called the cytoplasm. The process of DNA transcription occurs in the nucleus, as does the processing of hRNA to mRNA. The mRNA formed in the nucleus travels to the cytoplasm where the translation or protein synthesis occurs. Here is a summary of the transcription and translation processes in terms of the types of RNA involved and the cellular location where the processes occur. So next is the transcription or the RNA synthesis. Transcription is the process by which DNA directs the synthesis of hRNA and mRNA molecules that carry the coded information needed for protein synthesis. So it is a two-step process. First is the production of hnRNA, then second is the editing to yield mRNA. Within a strand of DNA molecule are instructions for the synthesis of numerous hnRNA and the mRNA molecules. During transcription, a DNA molecule unwinds under enzyme influence at a particular location where the appropriate base sequence is found for the hRNA, mRNA of concern, and the exposed base sequence is transcribed, and a short segment of a DNA strand so transcribed, which contains instructions for the formation of particular hRNA, mRNA, is called gene. A gene is a segment of a DNA strand that contains the base sequence for the production of a specific hRNA or mRNA molecule. So next is the steps in the transcription process. First is a portion of the DNA double helix unwinds, exposing some bases. The unwinding process is governed by the enzyme RNA polymerase rather than the DNA helicase. Then second is free ribonucleotides, one nucleotide at a time, and align along one of the exposed strands of DNA bases. the template strand, forming new pairs. In this process, U rather than T aligns with A in the base pairing process because ribonucleotides rather than deoxyribonucleotides are involved in the base pairing. Ribose rather than deoxyribose becomes incorporated into the new nucleic acid backbone. Then next is the third process is RNA polymerase is involved in the linkage of ribonucleotides one by one to the growing hnRNA molecule. Transcription. Then next is transcription ends when the RNA polymerase enzyme encounters a sequence of bases that is read as a stop signal. The newly formed hRNA molecule and the RNA polymerase enzyme are released, and the DNA then rewinds to reform the original double helix. So when compared to replication, you'll notice that it is quite different on some parts, because in the replication process, after unwinding the double helix and getting two template strands, the template strands will be used to make complementary bases and eventually the new strands. Then after finishing, the templates will remain a part of the newly formed daughter DNA molecule. But in the transcription process, you will see that after using the template strand to create complementary bases, the main thing here is that it will not result to new daughter DNA molecules, or it will not become a part of it as a strand, since it will just rewind back to its original double helix form and will release the newly formed RNA. So here is a diagram of the first step in transcription. Note that the direction of the hRNA strand after releasing is antiparallel so that of the DNA template. This will be always the case during transcription. That is why it is standard procedure when writing and reading the base sequences for nucleic acids to always specify base sequence in the 5 to 3 direction unless otherwise directed. So here is an exercise on the complementary base during transcription. So if a template strand of DNA is 5' CGAAT CCTA 3', what is the complementary RNA strand that is created from this template during transcription? So the answer here is letter C. However, there is a final form of the answer, which is this one, because the proper way to read or write of a sequence is by the 5' to 3' direction. So next is a post-transcription process, or the formation of RNA. The RNA produced from a gene through transcription is RNA, the precursor for mRNA. So post-transcription is just the conversion of hRNA to mRNA. And in this processing, certain portions of the hRNA are deleted and the retained parts of it are then spliced together. This process leads us to the concepts of exons and introns. It is now known that not all bases in a gene convey genetic information. Instead, a gene is segmented so that it has portions called exons that contain genetic information and portions called introns that do not convey genetic information. So a gene is simply a part of a DNA molecule that consists of alternating exons and intron segments. So both the exons and the introns of a gene are transcribed during the production of hRNA. The hRNA is then edited under enzyme direction to remove the introns, and the remaining exons are joined together to form a short-term RNA strand that carries the genetic information of the transcribed gene. So the removal of the introns and joining together of exons takes place simultaneously in a single process. The edited RNA produced is the messenger RNA that serves as a blueprint for protein assembly. So splicing is the process of removing introns from an hRNA molecule and joining the remaining exons together to form an mRNA molecule. So the splicing process involved snRNA molecules, the most recent RNA types to be discovered. Next is the alternative splicing. So the concept of alternative splicing bridges the gaps between the between the larger estimated number of proteins and the now lower estimated number of genes. Alternative splicing is a process by which several different proteins that are variations of a basic structural motif can be produced from a single gene. In an alternative splicing, an hRNA molecule with multiple exons present is spliced in several different ways. And in this picture, it will show 4 alternative splicing patterns that can occur when an hRNA contains 4 exons, 2 of which are the alternative exons. So next is the genetic code. The nucleotide-base sequence of an mRNA molecule is the informational part of such a molecule. This base sequence in a given mRNA determines the amino acid sequence for the protein synthesized under that mRNA's direction. This gives rise to the question, how can the base sequence of an mRNA molecule encode enough information to direct proper sequencing of 20 amino acids in proteins, considering that only 4 different bases are involved? If one base is only needed for encoding to synthesize a protein, then that would only result to a total of 4 amino acids. However, if 3 base sequences were used to code for amino acids, there would be 64 possible combinations, which is more than enough for uniquely specifying each of the 20 standard amino acids found in proteins. So research has verified that sequences of 3 nucleotides in mRNA specify the amino acids that go into synthesis of a protein. Those 3 nucleotide sequences we discussed earlier are what we call codons. A codon is a 3 nucleotide sequence in an mRNA molecule that codes for a specific amino acid. This gives rise to another question: which amino acid is specified by which codon, since we have 64 codons to choose from? After many experiments, it was found out that 61 out of 64 codons formed by various combinations of the bases A, C, G, and U were related to specific amino acids. The other 4 combinations were termination codons, or the stop signal for protein synthesis. Collectively, these relationships between the 3 nucleotide sequence in mRNA and amino acid identities are also known as the genetic code. The genetic code is the assignment of the 64 mRNA codons to specific amino acids or stop signals. So the genetic code has several remarkable features, and these features tells us that the genetic code is highly degenerate. Many amino acids are designated by more than one codon. Codons that specify the same amino acids are synonyms. 2. Genetic code has a pattern to the arrangement of synonyms in the genetic code table. All synonyms for an amino acid fall within a single box. Number 3 is the genetic code is almost universal. The same codon specifies the amino acid, whatever kind of cell it is, whether it be a corn cell, a human cell, animal cell, or other types of cell. Then 4th and lastly, the genetic code has an initiation codon. Besides coding for the amino acid methionine, the codon AUG functions as an initiator of protein synthesis when it occurs as the first codon in an amino acid sequence. So here is the table, the genetic code table. So here is another exercise. If a codon sequence of an mRNA is 5-GCC-AUG-GUA-UGC-GAC-CCA3', what is the amino acid sequence encoded by the mRNA codon sequence? And the answer here would be alanine, methionine, valine, lysine, cysteine, asparagine, and proline. So next topic is the anticodons and tRNA molecules. Note that when the encoding is finished, the amino acids used in protein synthesis do not directly interact with the codons of an mRNA molecule. Instead, tRNA molecules functions as intermediaries that deliver amino acids to the mRNA. At least one type of tRNA molecules exists for each of the 20 amino acids found in proteins. All tRNA molecules have the same general shape, and this shape is crucial to how they function. Here shows the general two-dimensional cloverleaf shape of a tRNA, a shape produced by the molecules folding and twisting into regions of parallel strands and regions of hairpin loops. Two important features of the tRNA structure are of particular importance. The 3' end of the open part of the cloverleaf structure is where an amino acid becomes covalently bonded to the tRNA molecule through an ester bond. Each of the different tRNA molecules is specifically recognized by an amino acid tRNA synthetase enzyme. These enzymes also recognize the one kind of amino acid that belongs with a particular tRNA and facilitates its bonding to the tRNA. Second is the loop opposite to the open end of the cloverleaf is the site for a sequence of three bases called an anticodon. An anticodon is a 3 nucleotide sequence on a tRNA molecule that is complementary to a codon on an mRNA molecule. The base pairing interaction between anticodon and tRNA and the codon of the mRNA leads to the proper placement of an amino acid into growing peptide chains during protein synthesis. An example, a tRNA molecule has the anticodon 5 AAG, 3', with which mRNA codon will this anticodon interact? Or what is the amino acid sequence encoded by the mRNA codon sequence? So the answer here would be 5' CUU 3L3', which stands for the protein leucine. Then the next is the last step, which is the translation or the protein synthesis. So, translation is the process by which mRNA codons are deciphered and a particular protein molecule is synthesized. The substances needed for the translation phase of protein synthesis are mRNA molecules, tRNA molecules, amino acids, ribosomes, and a number of different enzymes. A ribosome is an rRNA protein complex that serves as a site for the translation phase of protein synthesis. Here are the ribosome structure. It is said that it contains 4 rRNA molecules and about 80 proteins packed into 2 rRNA protein subunits, one small and one large. Then next is it contains approximately 65% rRNA and 35% protein by mass. Then another feature is that the ribosome's active site, the large subunit where amino acids are added one at a time for protein synthesization. Then next, number 4, it is said that the active site is mostly rRNA with only one of the ribosome's many protein components being present. Then next, number 5, it is said that the ribosome is thought to be an RNA enzyme that is a ribozyme. Then lastly, number 6, is that the mRNA binds to the small subunit of the ribosome. So, next is the translation process. First is the activation of tRNA. So there are 2 steps involved in the tRNA activation. First, an amino acid interacts with an activator molecule to form a highly energetic complex. This amino acid activator complex then reacts with the appropriate tRNA molecule via ester bond at its 3' end to produce an activated tRNA molecule. Then second step of translation process is the initiation. The initiation of protein synthesis in human cells begins when an mRNA attaches itself to the surface of a small ribosomal subunit such that its first codon, which is always the initiating codon AUG or methionine, occupies the site called P site. An activated tRNA molecule with anticodon complementary to the codon AUG attaches itself through complementary basing to the AUG codon. The resulting complex then interacts with a large ribosomal subunit to complete the formation of an initiation complex. Since the initiating codon AUG codes for the amino acid methionine, the first acid in a developing human protein chain will always be methionine. Then the third process is the elongation. The second binding site or E site in an mRNA ribosome complex is where a tRNA molecule with the appropriate codons binds to the next exposed mRNA. So, to simplify the process of elongation, the initiation tRNA carrying the amino acid number 1 is going to bind at the P site. Then next is a tRNA with amino acid to binds at the A site. Then next, a peptide bonds forms between amino acid 1 and amino acid 2 as amino acid 1 moves from P site to the A site. Then the first tRNA is released. The ribosome moves one codon to the right, translocating the dipeptide to the P site, and the tRNA with amino acid number 3 occupies the A site. Then the elongation continues as the dipeptide at the P site is bonded to amino acid at the site to form tripeptide. Then it will just continue repeating the process. Next is the fourth process, determination. So the determination is where the polypeptide continues to grow by way of translocation until all necessary amino acids are in place and bonded to each other. Appearance in the mRNA codon sequence of one of the 3 stop codons, UAA, UAG, or UGA, terminates the process. No tRNA has an anticodon that can base pair with these stop codons. The polypeptide is then cleaved from the tRNA through hydrolysis. Then the last process is the translation process. The post-translation processing. So some modifications of proteins usually occurs after translating. This post-translation processing gives the protein the final form it needs to be fully functional. Some of the aspects of post-translation processing are the following. First, in most proteins, the methionine residue that initiated protein synthesis is removed by a specialized enzyme in a hydrolysis reaction. A second hydrolysis reaction releases the polypeptide chain from its tRNA carrier. Some covalent modifications of a protein can occur, such as the formation of bisulfide bridges between cysteine residues. Then, lastly, completion of the folding of polypeptide into their active conformation occurs. Protein folding actually begins as the polypeptide chain is elongated on the ribosome. For protein with quaternary structure, the various components are assembled together. And with some recent research indicates that there may be a connection between synonymous codons within the genetic code and protein folding. It now appears that synonymous codons, even though they translate into same amino acids during protein synthesis, have an effect on the way emerging proteins fold into their 3-dimensional shapes. This means that 2 stretches of mRNA that differ only in synonymous codons can produce proteins with identical amino acid sequences but different folding patterns. Two differently folded proteins would be expected to produce different biochemical responses within a cell when interacting with other substances. There is some evidence now that this is the case. So that would be all of my video presentation. I hope you learned well. Thank you very much for listening. I'll see you in the next video.